All right, good evening, everyone, and welcome to Genomics Lite. This is our sequencing malaria session, and our guest speaker is Juliana Cudini, who's joining us from the Sanger Institute this evening. My name's Em. I work at Welcome Connecting Science on the campus, and I'll be sort of hosting and looking after the session this evening. Before I hand over to Juliana, I'll do all of the sort of housekeeping stuff that if you've been here before, you've probably heard me say before. All the sessions we host here are to help people explore what science means for them and for society. And so throughout the session, we do, ask that you ask Juliana questions about her career and what she does, but we ask that you do so respectfully. I'll be moderating the space this evening to make sure it is safe and inclusive for everyone. In case you somehow not used Zoom over the last couple of years, if you obviously can't use your camera or your microphone this evening, so if you want to get in touch with us, you can use the chat box or the Q&A box. At points throughout the session, Juliana will ask you questions and you can type answers in the chat box. And if you have questions for her, pop them in the Q&A box and I'll pick some out as we go and at the end as well. If you want closed captions this evening, they are available by the little CC button, and you can also move them around the screen so that they're never sort of in the way of Juliana's presentation. We'll also be using little polls that will jump up on your screen and give you a multiple choice question. I know sometimes if people are on a tablet or phone, you can't see them, so just type any thoughts or ideas in the chat box if that is the case for you. As I said, we are from the Welcome Genome campus, so Juliana is joining us from one of these buildings here this evening. And we've got lots of different science that happens on site all around genomics, which we'll be exploring this evening in terms of malaria. Some bits that might be of interest to you. We recently published 4 new pieces on RNA sequencing and bioinformatics on our website, yourgenome.org. And some of what Juliana will be talking about is linked to RNA and RNA sequencing. So these might be of interest to sort of wider reading. And as always, there's like a resource pack to go with this session. So if you want to learn more about malaria, you can go off and learn loads more about it and about single cell sequencing as well. Lastly, we've got a new series restarting in April called My STEM Future Speaker Series, which basically is here to showcase different scientists and different areas of science. This is available for anyone to join and to hear about different careers in science. Mariah works in our business operations team, so she helps science be translated into business, which is a pretty unique role. So we're really excited to chat to her about that. For this evening then, we've got our talk from Juliana. We'll follow that by a Q&A and we'll finish between 5:30 and 5:45. And this whole session is being recorded and will be available on YouTube and on our website after. So if you have to leave before then, you can catch up afterwards. I think that's all of my bit, so I will stop sharing and I will pass over to Juliana.
Okay, are we seeing the right view this time?
You just click the little swap one.
So there we go.
Awesome.
Okay, great. Welcome everybody. Thank you for coming. I'm really excited to talk about the work that I'm doing here at the Sanger Institute. I'm in the big long building that looks like a maze if anyone's been here. So I work on malaria. And I study malaria at a single cell resolution. That's kind of the tagline of my talk. And yeah, if you have any questions, please put them in the chat. I'm happy to stop and slow down and explain anything that kind of wasn't clear, because I think we're going to go through quite a lot of stuff that I work on this evening. So, or if anyone wants to contact me also, my email is down below, just give a shout during the question and answer session. Great, so, oops, all right, it's not switching to the next slide. 1 second. Just start on the next slide. Yeah, that's good. Okay, great. Sorry about that. Great. So I'm from Canada. You might have thought that my accent sounds American. It is not, gladfully. I am from Canada. I'm from this very, very tiny island in Canada. It's where I spent most of my summers. It's called Nova Scotia, specifically Cape Breton, and there's a whole lot of nothing over there. It is completely empty. It's completely just full of fields. And so I spent my childhood doing a whole bunch of nothing, and I was super bored. And that's when some scientists from the local university, Dalhousie, noticed that I was really bored and said, "Hey, little kid, do you want to get put to work?" And so I worked on looking at birds with them. So they were studying the evolution of birds and bird behavior and antagonistic behavior. So how birds basically fight with each other over territory. And so they got me to sit there and sit with a notepad and count how many times a bird came along, and hopefully the video will show, came along and kind of hit the other bird. There's a decoy bird there. And while that sounds really boring, it was kind of my first foray into science. I think before that point, I had no interest in being a scientist until I kind of got included in this study. And it's kind of why I like doing sessions like this, because it was really, really important to me that I had that kind of experience. And so it made me really interested in science at that point. So then that inspired me. I did my undergrad at, here we go, in this, I moved to a different area of Canada, did my undergrad at Western University in evolution. So at that point, I was looking at fruit flies and how they were evolving. I was kind of also looking at birds, looking at a whole bunch of different stuff. And I kind of got a bit sick and tired of doing the same thing where I go out and look at birds every day and sitting outside for long periods of time. And so I started looking at pathogens, so bacteria, parasites, viruses, because they evolve really, really fast, and you can see them under a microscope. So you don't have to travel out to the edges of the world to look at these things. You don't have to go out and battle the elements. You can sit in your cozy little lab and look down a microscope and look at them. So I followed that goal. This is very geographically out of scale. I'm very sorry about that. But I went across the ocean to the UK. Specifically to UCL to do my master's degree in pathogen evolution. Pathogen just being a fancy word for bacteria, viruses, and parasites. Anything that infects us and makes us sick, really. Couldn't decide what pathogen study. I've studied viruses, I've studied plenty of bacteria. And then I went to Cambridge to study malaria. So that's where I am now. And that will be kind of the focus of my talk. But I did want to highlight, throughout this entire period, I did not know I didn't really have a master plan. I didn't know I was going to end up at UCL. I didn't know I was going to end up in England at all. At each step of the game, I didn't really know what was going to come next. And so I like to highlight this because, if you looked at my degree and my career on paper, it looks really put together, but it wasn't. It was just me in the middle of nowhere being asked to look at some birds. And, then I just kind of followed what was interesting to me and ended up here. So I like to say this because even if you don't know what you have, if you don't know what you want to do and you don't have it figured out, that's okay. You can still somehow end up at Cambridge, maybe. So the focus of my talk will be on what I'm doing here. That's me in the lab doing sequencing of malaria. Not often smiles in the lab, but that was a rare good day. So we snapped a picture. And that's actually a picture of me sequencing the stuff that I'm going to show you in this talk. So that's kind of fun and exciting. So if we start off with malaria as a background, I know this is probably something that has been gone over in class, but I will give a quick introduction to malaria itself. For those of you who are unfamiliar, who fell asleep during the malaria section of class. So as we all kind of remember, malaria is transmitted by mosquitoes. And when a mosquito is infected or is carrying malaria, if they then bite you, they inject malaria into your bloodstream via that bite. Oh. Is that working?
Yeah, I think it was just going slowly, so we'll just give it a second.
Great. Right, so that injects into the bloodstream. Those cells are then going to travel to your liver, and they're going to infect liver cells. And then they grow and hang out in your liver for quite a while, and they replicate, and eventually they're going to burst out of those cells in your liver and enter your bloodstream. And that is where most of the symptoms of malaria happens. So they burst out and they infect an individual red blood cell. They then grow and develop within that red blood cell. And they go through this thing we like to call the asexual cycle. So they're replicating asexually, they're replicating exponentially. So they grow and develop within one cell, they replicate many, many times, and then they burst out of that cell. And then each one of those individual malaria parasites is then going to infect a new red blood cell. And that's how malaria grows within your body. And it's when they burst out of those cells that you get really sick, because as you can imagine, your red blood cells kind of exploding in tandem isn't necessarily good for you. However, importantly, these are not the stages that then go to the mosquito and transmit to the next person. So in order to do that, the malaria parasite has to leave this kind of cycle and commit to becoming what we call gametocytes, or the sexual stages. And that's kind of like the gametes that you have in your body, the male and female gametes that become eggs and sperm. This is just kind of the malaria version. So they differentiate into these gametes, and then those gametes, one male or gametocyte, one male and one female, get picked up by a mosquito, and then they replicate within the mosquito, and then that's what gets transmitted to the next person through the mosquito. So that's the very long and complicated life cycle of malaria. And there's a whole number of stages with when that's part of this life cycle. There's a whole bunch of different people working on different parts of this. And there's drugs you can give to stop the symptoms from happening, to stop that kind of explosion of red blood cells. But really importantly, one of the largest and most important, well, I say most important in a maybe biased way because I study these sections of research, is to look at malaria transmission blocking interventions. So those are drugs or strategies or interventions we can use to stop malaria parasites from transmitting between people. So it's kind of separate from the ones that stop you from getting sick. It's the ones that stop you from transmitting to other people. So I guess we'll have our first poll, which is just an open question. Where in this whole life cycle that I've just described do you think we might want to target if we're going to be stopping malaria from transmitting to other people, rather than stopping someone from getting sick? So that's the asexual blood stages, which is that circle I've showed here, the liver stages, so the replication within the liver, the gametocyte stages, so these kind of gamete-like organisms, or the stages within the mosquito.
So that's up on the screen now. Looks like most people are going for the bottom two, so the gametocyte and mosquito stages. A couple of votes for the other one, but gametocyte is winning by a little margin. So we'll give it another like 30 seconds. But yeah, gametocyte's still in the lead, followed by mosquito.
Well, that is great news. I'm a gametocyte biologist. I love to hear this. Everyone thinks what I do is important.
And that's most people voted in. Yeah, gametocyte at 55% of people, and then mosquito at 35%, and then a couple of votes for the other ones.
Yeah, that's great. That's really great. I think there is really no wrong answer. I mean, technically, stopping any part of this life cycle from developing will stop you from being able to transmit to another person. But yeah, you guys got it right on the money. The kind of last two stages, the gametocyte specific and the mosquito specifics, are really where everyone's targeting drugs for transmission blocking. So that's great. Yeah, so that leads us into exactly what I work on, which is developing these drugs that will stop this process. So stopping the cells from leaving the asexual cycle and developing into gametocytes, because if we can do that, we can stop them from getting to the mosquito in the first place. But there are lots and lots of interventions, bed nets being a really famous one that's trying to stop the mosquito from transmitting as well. So we can kind of wipe out mosquitoes, we can stop mosquitoes from getting infected with malaria, we can stop mosquitoes that are infected with malaria from biting people. But yeah, the focus of my research is stopping them from even getting malaria in the first place. So another maybe more challenging question, now that we know that we're kind of targeting the gametocyte specific stages, one of the big questions is, what if we're targeting these rather than those asexual cells? We need to know what kind of makes them different. We need to know how to target those specifically. And so can you think of any experiments we might want to do where we could kind of find out what makes gametocytes different from asexual cells? And there's no wrong answers, and this is very open. So put your answer in the chat box if you have any ideas about these.
We've got one idea that we could see what different proteins that are expressed in the gametocyte versus all the other stages, which is, I think, pretty much what you're going to be talking about in a second, which is always a good start.
Yes. Yeah.
Oh, this is an interesting one that I don't know much about. Identify monoclonal antibodies and look at antigens.
That's very interesting. Yeah, that's not the focus of what I'm doing, but that is certainly that's interesting because we have found that the proteins at monoclonal antibodies usually target the proteins at the surface of the cell, and it's really challenging with malaria because these cells are expressing the exact same things. They have the exact same genome, but they're so different across all these different stages, and so it's really hard to find reliable markers that really differentiate the sexual stages from the non-sexual stages, but we have found a few, and that's kind of even related to the protein question. If you can find proteins that are different between the two, and taking one step back, finding genes that are different between the two, then you can find potentially different proteins to target with antibodies. Those are both really, really good answers.
And then one more saying, a couple more coming in. So one saying, look at gene expression at the gametocyte stage. And then another sort of similar to that antibody one saying target different structures on the cell surface or different molecules expressed between the two. So yeah, I think generally looking at the difference between what the cell's up to at the asexual cycle compared to what it gets up to when it differentiates.
Yeah, that's great. My thesis title was basically gene expression differences on the gametocyte. So you guys, yeah, that is exactly it. That's really good to hear. It's very validating because, I'm glad I'm working on something that makes sense. Right. Yeah, I mean, that leads really well into exactly what I did. I looked at RNA sequencing. So I'm looking at, like I said, one step before protein expression. I'm looking at the genes that they're expressing. And so hopefully this is a bit of an overview of RNA biology and DNA biology. But in case, we're a little bit weak on the, or forgetting the central dogma of gene expression and genetics, every cell has the same DNA. In malaria, it's just about what cells are expressing what. So which instructions do you use determines kind of what cell type you're going to be, because not all cells are using all the same set of instructions, i.e. not all cells are expressing the same genes. And those genes are then, of course, going to be transcribed and translated into proteins. And then those are the workhorses of everything that's happening in the cell. So knowing what genes each stage is expressing is really important for us to know how we can target those with interventions, with drugs. And even before that, understanding the biology of what's happening at each of these stages. So in this kind of easy example, I've got two stages. I've got those rings. So that's the first stage right when they infect a new red blood cell as part of the asexual cycle. And then we've got gametocytes, and these are specifically male gametocytes. And you can see that in this example, I've got ring stage parasites are expressing gene A and gene B, and the gametocytes are expressing gene D and gene E. And this is just a little blurb on transcription in case you need a refresher. So when I'm looking at gene expression, I'm looking at specifically the mRNA that it's expressing. So this is after transcription. It's transcribed, the DNA is transcribed into mRNA that all have a very similar structure. They've got a 5' cap, which is just a feature of the mRNA, and they've also, all of them have poly A tails. That's a whole bunch of A, the nucleotide adenine added to the end of the transcript, the mRNA transcript. And that is something that does a whole bunch of things, but largely localizes it to the nucleus so that it can be read and translated and made into a protein. And we leverage this in order to capture it. So we have all of this mRNA, these genes being transcribed and translated within a sample, and I can extract that RNA using RNA extraction techniques. But how do I actually kind of pull it out, specifically the RNA, and sequence it? And to do that, we leverage the poly A tail. So another poll, and this one should be hopefully a little reminder of biology. What DNA nucleotide does adenine pair with? So I've got the other three up there. So do you remember which one pairs with adenine?
think I can safely say the answer is yes. We've got 98% of votes for thymine.
Good. Yes, it is thymine. And we leverage that by knowing that thymine is, sticky, it's attractive to the poly-A tail. And so we essentially fish for the RNA in our sample by looking at that poly-A tail, which should be on the end of all the transcripts. And of course, logically, we put in poly-T. We put in a whole bunch of Ts, and that should kind of allow us to clasp on to an mRNA transcript and pull it out of the sample. And we can start the priming of our sequencing using that poly-T. So that's the way that we fish in a sample and take out the RNA molecules from any stage. So in here, we can see we've got a representative of gene A, we've got a representative of gene D, and then we've got a second one of gene A. And so, at a surface level, I can say, oh, there's, a lot of gene A being expressed because there's twice as much as gene D. And that allows us to, on a much bigger scale, allows us to kind of create expression profiles, and they look like this. So gene expression is basically how many times we detect an mRNA transcript of the same gene. So if I'm seeing a lot of gene A in a specific sample, we're going to get, and this is on a color scheme, but it's essentially a count. So if I'm seeing, 15 counts of gene A, I found 15 molecules that I've sequenced that are gene A. We're going to count that as quite highly expressed compared to maybe in this sample, gene A is not being expressed that much. And so a lot of times we translate that into this kind of color scheme because it's a lot easier to visualize. And so in this case, a red value is kind of something that's very highly expressed. We've counted a lot of it. It's got very high gene expression versus a yellow or a white value that is not detected as much. And that's how we basically look at the difference between the gene expression between samples. So in this case, samples 2 and 3, I can likely say, based on the genes that they're expressing, which looks to be most highly genes D and E, based on my schematic over here, which is of course simplified, genes D and E are gametocyte male specific. And so that allows me to say, it looks like in samples 2 and 3, we've captured male gametocytes. Using that same logic, if I was to ask you, what stage do you think of malaria is most likely represented in samples 5 and 6? What do you think stage that is based on the gene expression profiles?
There's another poll on your screen now. And if you've got any thoughts as to why you picked your answer, you can pop those in the chat box as well. I'll give everyone a second to have a go at that poll. Awesome. So a little bit more mixed than the last ones, but we've got about 70% of votes for the ring stage and then a few votes for each of the others.
Okay. It is indeed the ring stages. The way that I figured that out is by looking at samples 5 and 6 down here. And if you follow that up, it looks like it's not really expressing gene F. It's kind of expressing gene E, not really expressing gene D, but it's really highly expressing genes A and B. And based on my schematic, the ring stage parasites are expressing genes A and B. So of course, these stages are expressing more than just 2 genes. They're expressing hundreds, in some cases thousands. And we're doing the same thing, just on a larger scale. So I make, you'll see later a heat map of gene expression that looks like this, but it's much, much bigger. And so we take all of the gene information and leverage that to find out what's happening in each of these stages. So in our little kind of mini RNA sequencing experiment here, what we've learned is that genes D and E are gametocyte specific and genes A and B are ring specific. So if I'm looking to knock out ring stages, I'm probably going to start looking at drugs that target genes A and B. If I'm looking to knock out gametocytes, I'm going to start to investigate genes D and E, understand what they do, what proteins are they at the surface, are they not? Because if they are at the surface, we can look at antibodies. And if they're not, how can we disrupt that biology? So yeah, I mean, that's what I just said. It's a little bit of a summary of this section. RNA sequencing is a really, really useful tool in malaria biology. And in, I mean, many different kinds of biology, it allows us to investigate what genes are being used during what stage in the life cycle. And then we do that by counting how many of those genes we are able to capture, the poly-T, and then sequence. So that's all great. My next step is to then, of course, look at the gene expression in gametocytes like we determined earlier. However, it's a little bit of a problem with gametocytes. And the problem is, how do we access them? So if you remember from the kind of summary of malaria, every single time that a malaria parasite goes through that asexual stage, it propagates and exponentially grows the number of malaria parasites in your body, because each one is going to infect a cell, go around that cycle, divide itself, sometimes up to 8, 10 more times. That's going to burst. And then every single one of those that all came from one malaria parasite is then going to infect a new cell, and that cycle continues. So each replication cycle, you get tons more malaria in the body, in the blood cells. And that's what makes you really sick. However, that same cycle, that cell going around, if it then chooses to go into the gametocytes down here, only 2 of them are made, or actually usually only 1 of them are made. And that is a huge difference from that kind of 8 to 10 to more that happens during the asexuals. And very importantly, once those cells are finished becoming gametocytes, they can't go back. They can never go back and become asexual cells again. So it represents basically a dead end for the parasite. So it's really, really important that they get picked up by the mosquito, because if they don't, the malaria parasites basically just spent a whole bunch of time and effort making these cells that have gone nowhere. They can't propagate within the body, and they haven't been picked up by mosquitoes, so they can't transmit it. So because of that, gametocytes are super rare. They only make them when they need them. And a lot of times they only make them when they can kind of sense that mosquitoes are going to be around. There's a whole bunch of kind of really specific, interesting biology that happens with gametocytes in kind of sensing when to turn on, when to commit. Because of that, when I take a blood sample from somebody that has malaria, over 99% are going to be these asexual blood stages. And if I'm really interested in the gametocytes, that signal is completely lost. How do I pick those out of the bunch? And specifically, if I'm going to be taking a RNA sequencing sample, so in this case, I've taken the blood, I've used RNA extraction to get the RNA out, and I've used the poly-T to capture it and to sequence it. And now we've got a whole bunch of transcripts, and I'm looking at the signal in front of me. Even if I was lucky and I picked up one singular little gametocyte transcript, if I'm looking at the expression as a whole of that entire sample, it's going to be completely averaged out. It's going to be completely lost. It's very hard for me to find that one transcript as a needle in the haystack. And so because of that, we use something called single cell RNA sequencing, which is a really exciting, I think it's pretty new still, tool to sequence RNA from an individual cell and be able to track back what cell that came from. So I'll explain that a little bit in more detail and how that's useful for malaria afterwards. But first, I'm going to explain the kind of logistics of single cell sequencing and how we actually capture single cells, because it sounds really nice in theory, but it's actually quite difficult to do because these cells are really, really small. So this is a kind of cartoon of what's happening within the single cell sequencing machines. Each of these tiny little channels is only the width of an individual cell. So they are microns long. They're very, very small. And in each channel, you have oil passing through that channel. And oil, as we know, when it contacts water, emulsifies, it becomes a droplet. And so we leverage that in order to have it form a little droplet. And within that droplet, it encapsulates a cell and it encapsulates a bead. And that bead has a barcode on it, which is a unique set of nucleotides, A, C's, T's, and G's in a unique combination that basically tags that cell. And it tags every single mRNA transcript we can get from that cell. And then we break the oil droplets apart and then we just sequence it like normal. But it means when I sequence it, I have that teeny tiny little barcode attached to each one of those transcripts, and that gives me information as to what cell it came from. And I'll show a very fun video just in one slide about how that works. So the way that this is often portrayed is that the way that we were doing it before, this bulk RNA sequencing, where you just capture kind of all of the mRNA transcripts we can get from one sample. So that's basically looking at all of the mRNA in the blood. That kind of averages everything out. And like I said, when you have something that's super rare, like gametocytes, all of that signal is lost. I probably won't see it because you're just basically taking an average. And the average is, the over 99% asexual transcripts, versus single cell where we can actually pinpoint, and that is of course, fruit salad. That's the whole thing. We're just going to average that all together. It's going to just be, this is the transcriptome of a fruit salad. When we use single cell, we can actually tag these cells with, or tag these genes with what cell they came from. We can then deconstruct in a really satisfying way. This is actually from a whole photo series of a guy who takes bulk things and he deconstructs them. So he has one of like an evergreen tree, and he takes every single tiny needle off and he lines them up. It's very satisfying. But with this fruit salad, it means that we can say this fruit salad is not just a fruit salad. It contains bananas, oranges, apples, kiwis, and a little whipped cream on top, or sorry, squirty cream, like Canadian British difference. So that is the kind of benefit of single cell, is that it allows us to really deconstruct what's happening in a system that's really biologically complex, with a whole bunch of different cell types that are all at different kind of frequencies and levels of expression and all that kind of fun stuff. So this might be a somewhat difficult question. There really isn't a right answer, because technically, this kind of system is really, really useful on almost any biological sample you can think of. But is there any kind of other system other than malaria that you think this kind of dividing into its individual components would be particularly useful? Any systems that you think would be really cool to apply this to? And there really isn't a wrong answer, but just put your answers in the chat if you have any thoughts.
I had an immediate one for circulatory system. So I think that'd be really interesting, because obviously that's quite a big, that's a really big system.
Yes,
so
yes, that's a very good answer.
Likewise with immune system. Someone's put dengue with a question mark, so I'm not sure if they're not sure, or if you know anything more on that. Cancer cells, I guess this one's a really good one for being able to separate sort of healthy cells to sort of cancer cells, and someone said tumors as well. So I guess being able to take a tumor and look at the difference between cancer cells and healthy cells. Someone's put CRISPR-Cas9. I don't know if this work you can do alongside CRISPR.
Yes, actually, that's a super cool new area of research, is single cell CRISPR.
Wow.
So you can actually look at how a knockout perturbs a system. So what effect does taking that gene out have on the rest of the genes in that cell, literally at a single cell level? It's very, very cool. I would say it's relatively new, but that is very good forward thinking. I wish I could do that in my PhD. Yeah.
So someone's come up with their own thesis in the chat by the looks of things.
Oh yeah, I mean, this is great.
Someone else has put surface proteins expressed in cells along the gut. That's very specific. Someone's put so we've had a couple of ideas for HIV and AIDS. So again, I guess that comparison of healthy to unhealthy or disease state cells. Yeah, I think that's most of them. So yeah, some really, really good ideas and really specific ideas as well.
Yeah, those are all really great, and I think it highlights the like the dengue and the HIV and malaria. Those are all, I mean, both dengue and malaria are single-celled parasites. So the whole parasite is just one individual cell, versus humans and other organisms that are multiple cells types and multiple cells. So single cell is really useful for that, right? Because anything else is averaging. Even if you were to take a very small sample, it's still averaging a whole bunch of HIV, dengue, malaria, viruses or parasites, depending on what you're looking at. So single cell is super useful for those. As well as tumors, you can have sublineages of tumors. You can have tumors going off and doing different things that can kind of almost propagate within your body, almost like a little virus. The surface protein one's super interesting. We have some techniques. It's called site-seq that tries to tag an individual protein, and then you can do that at a single cell level too. So you can look at the surface proteins that individual single cells are doing. And yeah, the CRISPR-Cas9 thing. So those are all kind of single cell plus. So you've answered the question and you've gone beyond. That's great. But what I was thinking of when I wrote this question is actually the immune system. I wouldn't say it's the best. There's tons of systems that work well with this, but the immune system's been really, really useful to study with single cell, because as we know, there's just so many different components to the immune system, and different cells are going to react to different treatments in different ways, and you can infect So some people are actually looking at both malaria and the immune system cells that they're interacting with in the same kind of single cell experiment. And you can find different cell types of even, B and T cells that are attacking malaria in different ways. And I think that's really, really cool. Some people are doing single cell of immune cells in the mosquito when they are picked up or when they have malaria in them. So we can really dive down with single cell sequencing, and it's a super useful tool. So as promised, the cool video of how single cell works. This is an actual video of not my sequencing, but sequencing I did with one of my friends. And so if you remember that cartoon I showed of the different wells and the oil droplets, that's what you're seeing here. So I'll let that one run. It's kind of satisfying. So you can see oil droplets formed, and they're forming because we're pumping through not pure water, but a water-like solution to form these emulsified bubbles. And within that bubble, so coming from this channel up here, up at the top, we've got that big circle, and that big circle is the bead. So you can see the beads coming through and being encapsulated in a droplet. And then very small, and it's very quick, there's a cell coming from this channel, and it's really, really tiny. And it just kind of goes and it zips right through, and that is encapsulated in the droplet as well. So that is how single cell sequencing works, and we do basically the whole RNA sequencing reaction within that individual little bubble of oil, and I think that's very fun. So essentially, how am I? So now we've got the technology I need to find gametocytes. And I'm interested in the malaria parasite. So how does this work? How do I set up a single cell experiment? So all of this is done in the lab. I grow malaria in culture flasks in red blood cells. And if you guys have any questions about that, please feel free to ask how I do that. But when it's time to take a sample, I get my little collection of blood that contains hopefully a whole number of stages, even if it's not within the body and it's within this flask, it is still quite rare to find gametocytes. So this sample often contains a whole number of stages. So these 3 are from the asexual stage, and then we've got the 2 gametocytes down here. But the important thing is that each one of them has a tag, and that tag is that barcode that I've added to their mRNA. And that tag is the same for all of the transcripts we're getting, or genes that we're getting from that cell. And so now, instead of averaging it all out, we have this kind of information at our fingertips. So we can make the same gene expression kind of heat map, and so we've got the genes lined up along the side here, and instead of these being a whole sample of blood, it's actually an individual cell. So this is the tag, that pink tag here, and that's coming from this stage, the ring stages. Then we've got another cell, and so these matrices are huge. We've got hundreds and thousands of cells, and I've got hundreds and thousands of genes, and I can make pretty heat maps like this based on individually how much is gene A being expressed in that one individual cell. And so that's super useful, and I get it's so much information actually that it's really difficult to work with and narrow down on. So we have to do something that I think this can be a little bit difficult to explain. So please put your hands up or ask me to slow down if this is a little bit confusing. But I think it's super important to talk about because it's a really big up and coming field in biology as a whole, really. And I think it's a super interesting look at combining computer science and biology. So I'm, of course, a bioinformatician, and that is just one role as part of this whole thing. You can come at this from the biology side, you can come at this from the computer science side. So it kind of opens up a whole bunch of avenues. So you don't necessarily have to be the one that makes these samples and grows up these cells. You can actually just get the data at this stage because it's so big that we need lots of people to work on it. I mean, it took me 4 years of working on this one basically data set to write up into my thesis. So it takes a really long time. So this is a whole field of career trajectories if anyone's interested. And so this is a simplified kind of version of it, but in a sense it's called dimensionality reduction. It is basically just squishing the data down into something that's a lot more understandable. Because if I was going to get this huge matrix of all of the genes and all of the cells and all of the numbers of each of that gene. And in that cell, that's too much information to be able to process. It's really hard to narrow in on what's actually important. What are we learning from this? And remember, our overarching question is what genes are being expressed in what cell? How do I get at that? And especially like, how do I plot that? You know, I've got tons and tons of dimensions. I've got, in this case, I've got 30,000 cells times 5,000 genes. So how are you ever supposed to plot that? And that's done using a whole bunch of different techniques. But essentially, it's trying to boil this down into the most informative information. And we do that by trying to cluster cells. So each of these dots on this plot, well, actually, let's start with this top one. Each of these dots is an individual cell. And we draw a line between them whenever they share a gene in common. So between these two cells, they have a similar level of expression of the same gene. So we draw a line between them and saying like, there's something between these. They're kind of somewhat connected because they share this gene. In this case, we've got two genes that connect these two. So that means they're more related than the ones that only share one gene between them. In this case, these two cells are sharing the expression of 4 genes. So that then brings them closer together on this plot. And that allows us to say there's probably something that's quite similar between these two. If we were to examine those 4, say this is not malaria anymore, it's a human cell, and we're saying, all 4 of those genes are involved in heart function. It looks as though these are probably cells from the heart. And because they share so many genes, they're probably quite similar and doing very similar things within the heart. Because ultimately those are going to become proteins and those are going to be doing things. And so if they are expressing the same proteins, they're probably doing very similar functions. And that allows us to draw these huge graphs where each dot is a cell, and the proximity of that cell to another cell is equivalent to how many genes they share or how similar their expression graphs look like this. So I hope that's not too kind of abstract of a concept. Just to see how everyone's feeling, I've got a question, which is, I've been told this is not light blue, it's kind of a green. So think of this color as however you wish, but this cell specifically, in this plot, this little simple plot I have here, what cell is the most similar to that one?
Someone's put the light blue cell, but I think that might just be the one next to it because the colors are quite similar.
Yeah.
Yeah, someone's put the green cell, which I think is the one, yeah, dark green. Yeah, that one there.
Yes, I'm actually just realized, I'm very sorry to anyone who's colorblind, and I've now asked you to identify a cell by its color. You can also, the answer is the green one or the cell that is right beside it that shares the 4 genes with it. That's great. Yeah, so that is the principle behind this dimensionality reduction, and the kind of idea of how do we take data that's super informative, how do we mine data, how do we squish it down into things that make sense, and how can we squish it down and then expand it back out into things that are interesting? Can we squish it down and then use that information to predict what a cell type's going to be like? If I was to say, can I maybe use machine learning to predict what the expression profile of this is going to be? We can kind of use this kind of squishing and expanding of information. So that is essentially what I do. I spend a lot of time on the computer. I am very glad that I don't spend that much time in the lab, and I just take this data and squish it down in different ways and see what comes out. But this is really useful for us to answer that overarching question of what's different between gametocytes and asexuals. Because here you can see, because these cells share so many genes, and these cells share so many genes, but there's not that many ones shared between them, they actually cluster out separately. And that's what's happening here. And this happens a lot in human cells, where you have very specific subsets of things being expressed in very specific cells. But that doesn't always look like that in malaria. So I will show you what this actually does look like when you single cell sequence malaria. So I've put on the side here the life cycle to remind you of what that looks like. And this, ta-da, is the graph that we get if we single cell sequence malaria. So every single dot is an individual cell, and where those dots are on this graph is based on that principle we just talked about before, which is how many genes they share. So to talk you through this, because I was very excited to find this when I squished the data down in just the right way and got this. So these are the asexuals. You can see it forms that ring. So here we've got the ring stage parasites in greens. That's these boys right there. And then we continue around that cycle. We've got our trophozoites. So that's kind of the next development. So those ring cells have infected red blood cell. Now they're kind of growing, expanding. And then remember, they start replicating themselves into these called schizonts. So there we go. Those are our schizonts. And then those all burst and they infect new cells and they become rings again. So we've really nicely captured that whole cycle in an actual circle when we look at what genes are being expressed similarly. And what I said before about kind of these distinct clusters, malaria, because it's developing in this continuum. So there's not really, you don't become a ring and then all of a sudden jump to becoming a troph and then all of a sudden jump. There's DNA replication, there's stuff happening in between. And so whenever you pick up an asexual parasite, it's going to be somewhere along that circle. And so it's really nice to see that these cells, when we cluster them, they are kind of sitting exactly along that circle. And so if you were to take a cell here and a cell here, they're going to look very similar. There's just going to be a very few genes in between them. And those genes are going to be responsible for like that increased development time. So that was very exciting to see. But obviously we're looking at gametocytes. And so super exciting was to see the gametocyte stages represented on here kind of exactly as you would expect. We can see them leaving that asexual cycle. And these are intermediate stages between going from here to here. And then they differentiate into males and females. And those are these late stage females there, or late stage gametocytes there. So in this single cell experiment, I've managed to capture all of the asexual cycle and all of those cells that are leaving the asexual cycle and heading into gametocytes. And so at any point along here, I can start to interrogate what genes are they expressing and how can we knock them out? Because if we were to knock out genes here, we might stop them from becoming gametocytes altogether. If we were to knock them out here, we might stop them from becoming males. If we were to knock them out here, we might stop them from becoming females. Or knock them out maybe here. Maybe this is something that's more accessible. Maybe they're expressing different surface proteins. So it's really, really useful to have this information of kind of progression along that cycle. So what do I actually do with the single cell data? I've got it. How do I look at it? So this is actually a plot directly from my thesis. So lucky you see it before it gets published. And I like this plot because it shows really nicely, basically a giant version of that gene expression heat map we were looking at before. So in this case, in this smaller version, we're looking at genes along the side here. Along the y-axis, we have the same, it's also genes, it's just thousands of them rather than 6 of them. And along this axis, the x-axis, we have malaria parasites as they progress along that asexual cycle. So we've got this color map here. So I'm basically taking that circle and breaking it up into a line. And it gets a little yellow mark if there is expression of that gene in that part of the cycle. And it's kind of more simple to look at it this way. I can find a whole host of genes that have their kind of peak in expression right here in the ring stages, and then they progress on to trophozoite stages and we get another little peak. And then, it gets later in the trophozoites and we get another little peak. And then those schizonts, which are the replicated versions, and then very late schizonts right before they're about to burst and become rings again. So this was super informative. And it was the bulk of my research, which is essentially if I was to cut out a section of this, what genes are they expressing? And you can see the malaria parasite is very specific with what genes it's expressing and when. It really has a very set kind of progression of what genes is expressing in each stage. And that's super useful to us because then we can start to knock them out. So the next thing we can do with single cell data is exactly that. We can knock genes out This heat map is from the asexual cycle, but of course we're also interested in the sexual cycle, and I can make the same plot for the sexual cycles. It just wasn't as pretty, so I didn't put it in. But that's essentially what we did. In this case, this is a very similar plot. It's just showing the females and the males colored in green and red here. And based on these genes, we selected a few. This is work done with another PhD student in the lab. His name's Andy. We selected a few of these to actually knock out and see what happens. So in these gray plots on the side, we've got the healthy cells in gray. So you can kind of just see where the males and females are. And then anything in blue is one of these knockouts. And then each individual one of these is a different gene that we've knocked out. So you can see really well in the males, for these 4 genes that we've chosen, when we knock out that gene, we do not get any males ever being formed. We only get females being formed. We don't get any males. So that was really good. Those are really good targets to knock out with gene targets or other kind of interventions, because when you knock out that gene, it's not present, males aren't able to form. We saw something similar in females. They still were able to make a few females. You can see these kind of dots, but you can see a lot of them, they're really decreased in the number that they were before. There's obviously a lot of males being formed and not that many females. In this case, in this gene, they were able to form females, but they weren't able to kind of progress along there. So they started making early females and they didn't progress all the way to the end. So we're doing a lot of work with those gene knockouts. And lastly, we can actually bring this to natural infections. So what I said was a lot of this work is being done in cell culture. So that's malaria that I'm growing in the lab in red blood cells. But obviously we're interested in how these parasites look at a single cell level from actual patients, people who are infected with malaria. So we brought the 10x machine, the single cell sequencing machine down to Mali. That's a country in West Africa. It was actually the first time anyone's been able to get a single cell sequencing machine in West Africa. So we were super excited to do that research. And we worked with our partners in West Africa to collect samples. And that's me looking at some malaria from patients in Mali and working with this is the rest of us from the Lonazak lab at Sanger doing our work in Mali. And we were able to get single cell sequences from the infections of these patients. And so that allows us to find out if these knockouts are working in actual patients. Is this just specific to stuff that we're growing in the lab, or is it actually useful in natural infections? And then also, what's different between the expression? If I was to make this heat map for expression profiles of natural infections, how would that look different from the heat map of profiles in the lab? Sometimes they're expressing different genes, and very importantly, they have very different immune responses. So how is the malaria parasite responding to that? So we get really good information from that. So we're super, super excited to see what comes out of that study. So with that, I'd like to say thank you very much for listening. I think it's a good time to head into the question and answer section. If there's anything I haven't explained well enough, especially that kind of dimensionality reduction stuff, let me know. And yeah, thank you for listening.
Awesome. Thanks so much, Juliana. I always learn loads in these sessions. We've got a couple in already, but if anyone's got any questions, you can pop them in the chat box or in the Q&A and we'll pull some of them out. So we had one question as you were going through, how are the tags attached to the mRNA molecules when you're doing single cell sequencing?
Yeah, that's a good question. It requires a little bit of knowledge about how sequencing works. So when you're trying to get something to sequence, you have to add something that recruits the ribosome to actually start transcription. And we use that, it's called a primer sequence. And so it kind of, the primers that we use in, the ribosomes that we use in sequencing recognize a specific primer sequence, and that kicks off the transcription of the second half of the mRNA. So the poly A tail that we're using to fish out the mRNAs is attached to a primer sequence, and then that continues on and sequences the rest. And so that basically the primer as a whole is comprised of poly T tail, the primer, and that tag, the unique barcode. And so it will sequence through that whole thing. and it will continue on into the gene as well. So we get the information about the gene, and then we get that barcode, and then we get the poly T, which we cut off.
Awesome. It's just it's such a clever way of doing, like, sort of utilizing what the gene already looks like to sort of hijack it for, for your own purposes.
It is, and it looks very cool because the bead itself, it looks like a little hairy spider thing, actually, because each bead has hundreds and thousands of these tiny little poly Ts coming off of them with a primer sequence. And so what it means is that within your oil droplet, you have your individual cell, and we add something to burst that cell to lyse it so that all of its transcripts are coming out. And they're literally going to be then attracted to those poly Ts that are kind of stuck on this bead. And so eventually you're going to have a bead that just has a whole bunch of mRNA attached to it. And I think that looks very cool under the microscope.
Awesome. We've had one question about, you mentioned you were going through that you use blood in the lab to, like, grow up your malaria. So someone said, what type of blood do you use? If it's human, where do you get it from?
Yeah, that's a good question. We get it from the NHS. It is human blood. Malaria parasites, at least the ones that we work on, are human specific, so we have to grow them in human blood. But there's tons of other malaria parasites that infect a whole bunch of other species. But we have to use for falciparum, which is the most common and most lethal human malaria. We use human red blood cells, so it's specifically just the red blood cells. We wash out any white blood cells, and those are the kind of parts of the immune system which could kill our malaria. And because we're trying to actively grow it, which seems counterintuitive, we're trying to keep malaria alive, we wash out the white blood cells. So it's just red blood cells, and they are from patients that are donating blood to the NHS. And so what happens is, if the blood that you've donated isn't of good enough quality because of whatever reason to give to another patient, it goes to research. Or if it was not used and it was kind of left out a little bit too long to give to a patient, it also goes to research. So it's kind of a bonus if you donate blood. You could either save a life or contribute to scientific research. We specifically use O positive blood because basically malaria parasites can get used to a certain blood type when you continually culture them in the same blood. And I don't know who the first person was to use O positive. It is the most common blood type in many European populations, and so it probably was just availability. But all of our parasites are so used to using O positive that if we were to switch it, they would get upset. So we have to use O positive blood from the NHS, and it gets donated to us.
Do you think you could compare then if you grew up some in a different blood type and then did single cell sequencing? Do you think they'd be?
Oh, you definitely could. You definitely could. You would just get the transcriptome of a very angry, upset parasite. It's like, what are you doing?
Yeah. Love the idea of being able to tell if it's angry. Have a couple of really interesting questions. So someone said that their dad had malaria a few years ago. So if his cells were studied, would they still find malaria or like traces of having malaria in his cells?
Yeah, that's an interesting question. Largely no, especially in his blood, there should not be or would not be any more malaria in his blood. Where you might find it is in the liver. Sometimes, and particularly different species are more or less likely to do this. They can go dormant in the liver, so they can hang out in the liver without actually causing an active infection. They're just kind of lying dormant in there. So if they were to study a lot of his liver cells, I think it would be quite rare to find. So you basically have to get a whole bunch, and you'd have to single cell sequence it because it's so rare. You might find them in the liver, but yeah, if it was, depending on what kind of malaria it was, which was depending on where your dad was, if it was kind of sub-Saharan Africa, it was likely Plasmodium falciparum. If it was Southeast Asia, it might be some of the other species. But yeah, likely it won't be found actively replicating in his blood.
We've had a really interesting question of, is there a way, anything that scientists could do to promote differentiation into gametocytes to increase RNA yields from them? So trying to solve your problem from a different angle.
Yeah, that's essentially what people were doing before single cell. So that's a great question. There are ways that I grow parasites in the lab that promote gametocyte conversion. One of them, which is kind of funny, is to make them really stressed. So if you stress out the parasite, it's almost as if, they're within a host and they're saying like, "Ooh, it's not looking good in here. I'm within Julie, and she's getting really sick, and like, I'm not convinced it's a good idea to stay here. Let's jump ship." So if they're in an environment that they signal is not really fit and not really good anymore, they will increase their conversion to gametocytes and then try to run away. And so I simulate that in the lab by really stressing them out, because malaria parasites will keep growing and growing and growing and growing if you let them. They won't stop. They don't know what's good for them, especially in culture. And so I have to continually keep them low, or else, they get upset. But if I let them go high and I let them keep growing and growing and growing in the red blood cells, and I don't give them any more food, so I don't give them any of the nutrients they need to grow, they start to signal, ooh, this isn't a good host. Let's start to leave. And so they'll make more gametocytes. And so I just basically, it's easy for me because I can just leave. I can leave my cell culture and go away and have a coffee and do whatever I want. And they're basically within there replicating, getting really, really stressed, and they have tons and tons. So they sense how many, or they potentially sense how many parasites are around them, or nutrient deprivation, and then they will create more gametocytes. That's a way that I can enrich for gametocytes. There's also a really cool technique. It's called MAX, which is a magnet assisted cell sorting, I think. Don't quote me on that, but I think that's the acronym. But it is essentially using a magnet. And so there's different levels of different proteins and compounds within these cells, and specifically something called hemozoin, which is trying to break down the heme, which is part of hemoglobin in your red blood cells. And that is full of iron, so it's very metallic. And so different stages will have different amounts of this hemozoin. And you can then use a magnet to pull out specific stages, and gametocytes are particularly enriched. So you can use a magnet to separate them out and enrich that way. And that is a really good way of doing it. The important thing, though, is that really annoyingly, there are no sex chromosomes in malaria like there are in human gametes. So the only way, they all have the exact same chromosomes. So the only way that we can tell a difference between a male and a female is by what genes it's expressing. So at the moment, there's not a very good way of separating out male and females. Even if you're to enrich for gametocytes, you can't necessarily separate out males and females. So single cell is really useful for that. But yes, there are definitely hacks you can do to enrich gametocytes.
Like the idea that you've got lots of like stressed malaria, you've got lots of unhappy malaria in the wrong blood. You just spent 4 years aggravating malaria.
Yeah, I did.
Someone's asked, in one of your slides, there seem to be much more female gametocytes than male ones. Why do you think that is? And does that have an effect on like the knockouts being more successful in one than the other?
Yeah, that's a very good question. And I don't actually have an answer for it. It's something I'm actively trying to answer right now. But I can answer the first half of the question. There is something called the sex ratio in malaria parasites, and it's a very active field of research, understanding how many males and females are being made. So one cell can differentiate into a female, or it can differentiate into a male. And then they're called gametocytes. And then when they get in the mosquito, they then turn into gametes, just like we would know, the male and female gametes in the humans. And one female gametocyte will make one female gamete, but one male gametocyte can make up to 8 male gametes. So it can actually replicate and split up and make 8. And so what we're measuring is the step before that, before it makes 8. But when you think about it, it's actually really interesting in terms of an evolutionary sense, because if you assume that a malaria parasite is going to be picked up by a mosquito, and there's going to be no other malaria in that mosquito, it's just you've just made it into that mosquito. You only need to make enough females for those males to fertilize. So usually, you'll make a certain, however many females you want. So you can actually make more female gametocytes and fewer male gametes, because all of those gametes are going to become more male. Sorry, I did the other way around. You can make fewer male gametocytes, because those are going to make a ton of male gametes in the mosquito, and then all you need is enough to fertilize those females. So oftentimes when we pick up malaria, it has a lot more females than males. What's interesting is that sometimes a mosquito can pick up multiple kinds of malaria. So it can kind of, it would bite me, and then it would get my malaria, and then fly over to M and bite them, and then get a whole bunch more malaria. And it could be a different strain. So like variants, kind of like COVID, it can get different kinds of malaria. And in that case, if there's more females sitting around, and if you've accidentally made some extra males, those males can then go fertilize with those females. And then you've, kind of sneakily slid more of your genes into the next gene pool. And so it's a very interesting field of research is, in what context do gametocytes commit to becoming females or males based on what they think. So in areas that have really high transmission tend to have a lot of those mosquitoes biting multiple people. And in those cases, they actually have many more males than females because it's more evolutionary advantageous to have a lot of males. But yes, in those knockouts, why are we kind of completely getting rid of males and not necessarily always getting rid of females? We don't know. We don't know what the mechanisms are. It could be that, kind of similar to where how gametes form in the humans, you need signals to, everything is default female and you need signals to turn males on. That might be the case in malaria. We don't know. We're still actively investigating it.
That's so interesting. Cool. We've had another great batch of questions. I want to go through and pick them out. So someone's asked, do you ever use methods such as reverse transcriptase or using restriction endonucleases and or making it from scratch with a codon and bead, or does that not apply to malaria?
All of those, some of those are part of the sequencing process. So reverse transcription will happen because we are taking RNA during sequencing. We're taking the RNA that we're capturing and actually converting it back into DNA to sequence it. And so in that sense, we are using reverse transcription. When we sequence them, we can't sequence necessarily the whole length of the transcript. So sometimes we do have to cut them up. I'm not sure if that's always with endonucleases, but sometimes in some settings it is. It was something about a bead. I forget the last one.
Making it from scratch with a codon or a bead, codon and bead. I'm not sure what that.
Yeah, I'm not sure. Not sure about that one. But yeah, I guess the biochemistry of how sequencing works, by and large, is the exact same way between any kind of cell, human cells and malaria cells as well.
Awesome. I'm also a broad question. Someone asked, is the malaria life cycle similar to any other parasite, or is it fairly unique? Because it has got quite a weird life cycle.
It is. Yeah, that's a very good question. Yes and no. So specifically Plasmodium falciparum, so that's the one I study. It's the one that infects the most people, kills the most people. All of the Plasmodium parasites have a very similar, so all of the malaria parasites have a very similar life cycle. The length of time it takes to make those gametocytes differs a lot between different species. Weirdly, falciparum takes 2 weeks. The whole life cycle usually takes only about 48 hours. So in 48 hours, that's why people tend to get sick every 2 days. That's when all of those red blood cells are bursting and then they reinfect. And so that causes those like very easy to spot cyclical malaria fevers. But for whatever reason, even though that only takes 48 hours, the development into the gametocytes takes 2 weeks. And at the end of those 2 weeks, if that doesn't get picked up by a mosquito, then that was such a waste of time. So it's baffling still. I got asked it in my Viva, why do you think this happens? And the answer is we have no idea. We've known about malaria for hundreds of years, and why do we still not know this? But outside of Plasmodium, so outside of malaria, there are a few related parasites, and that is one of them is the most famous, I guess, is toxoplasmosa. And so toxoplasmosis is the disease caused by the toxoplasma parasite. That is often transmitted. It's most concerning in pregnant women. It's transmitted actually through cats and other mammals. So they often tell women who are pregnant to stay away from their cat's litter because it can be transmitted through their poop. And it's one of the only parasites that can cross the placental membrane, and so that's why it's so dangerous. But yeah, that causes toxoplasmosis. That is somewhat related to the malaria parasite, has a kind of similar-ish life cycle. It goes through an asexual replication cycle, and it also differentiates into kind of gamete-like things. So yeah, that one's close. And then also something called Cryptosporidium, which is a long word, and it's a parasite that infects the gut and can make you feel ill. And it's largely, I think, in Africa, but I don't remember if it's expanded past that. But those are 2 other parasites that have similar-ish life cycles.
So I'm going to pick up two more before we wrap up, and one that I'm really pleased someone asked, because if they hadn't, I probably would have asked myself. You sort of mentioned this idea of like mosquitoes being able to, not mosquitoes, the malaria parasite sort of maybe being able to sense if there's mosquitoes around, or like, as you said, getting really stressed to like decide to become gametocytes. So someone said, like, can they sense mosquitoes, and what's the process, like the biological process that they undergo to become a gametocyte, which I imagine is quite complex. Whether there's anything you want to say on that?
Right, okay, I'll take that in stages. Yes, we believe that there is some kind of, whether or not it's sensing or it's just good timing, we don't know. So we don't know actually how the parasite can potentially sense these things, but there is some evidence to say, it's actually a lab in Edinburgh that does a lot of work on this, which is, can this parasite sense when it's night and day? Because depending on where you are, different species of mosquitoes are either kind of day biting or night biting mosquitoes. And you'll see malaria parasites peaking. So I said there was that kind of like 48-hour cycle. We'll see the peaks of when gametocytes are detected, coinciding with when the local species of mosquitoes tend to bite. So if they're night biting mosquitoes, you'll see lots of gametocytes in the evening. And it's unclear if that's sensing or if that's just over time it's been selected for, that those parasites tend to start that process such that they'd be gametocytes by nightfall. So that's super interesting. There's also some evidence to say that males and females will kind of stick together and form in pairs and travel together in pairs, so that they're more likely to be picked up as a pair, because that's really important, because if you only pick up a female, it's kind of useless, or a male. And then there's also evidence to say that they prefer to be found in kind of small capillaries that are found at the surface of your skin. So obviously when a mosquito bites you, they can't go all the way through to, the middle of your arm. They're going to be picking up kind of at the very top of the dermis of your skin. And so there's evidence to say that gametocytes versus malaria parasites, the asexual parasites, gametocytes tend to form in those kind of skin capillaries rather than, or circulate in the skin capillaries rather than in kind of deeper capillaries. So that's really interesting. How a gametocyte forms is the whole basis of my PhD. So that would take a long time to explain. But there's a few stages. They go through actually 5 stages of development. First, they form within the blood, and then very quickly they leave the blood because they don't want to be picked up by the immune system while they're developing. And they actually travel into your bone marrow, and they form in your bone marrow, and they kind of hide away there for most of their development. And so that makes it an additional kind of challenge for me to find them from a patient, because a lot of times if I'm going to take their blood samples, the gametocytes aren't there, they're hiding away in the bone marrow, and it's very tedious and difficult to get a bone marrow sample from somebody. That's not fun. So that makes it hard because they're literally hiding from us. And then once they're ready and they're formed and they're in their kind of late stage mature male and female forms, they then leave the bone marrow, because of course they need to get picked up by a mosquito, and they travel around to those capillaries we just talked about. So those are the general stages, and they use a whole bunch of different proteins to do that. Interestingly, sometimes they recycle. So if a male needs to replicate into those 8 different gametes from a gametocyte, they use very similar proteins and mechanisms that they used earlier on from the trophozoites replicating to become schizons and then bursting. So there's a whole host of biology happening as gametocytes develop.
Awesome. And then a really quick last one. Someone's put sort of, what's the end goal of your research? I'm guessing it's to do with treatment, but yeah, what would if someone picked up your thesis, and what would you hope they did with it?
Burn it. No, I'm kidding. Okay. The end goal of my research is, trying to understand biologically what's happening as these gametocytes develop. I mean, it takes a lot of time and effort to make actual drug targets. So I think, of course, the end goal would be translational drug targets, but that's a little bit down the line. First I need to show them the targets, then we need to knock them out, and then we need to find out, okay, we know that this gene is important when we knock it out, let's find drugs against it, and then you have to trial all of those drugs, and then you have to narrow that down, and that might become a treatment. But that is essentially the end goal. But then also, just in terms of basic research, understanding how the biology of what is happening and what genes are being used and how can we disrupt things.
Oh, I've lost your audio. I can't hear you. Don't know if that was just me.
Oh, can you hear me?
Yeah, I just lost you for a second, but it might have just been my speakers.
Oh, okay, cool. Sorry about that.
No, it's fine. I think we'll wrap up there anyway. Thank you so much for joining us and like answering so many of those awesome questions. I'm just going to share my screen very quickly to wrap us up. If you've enjoyed this session, we are doing this sort of monthly, so our next one will be at the end of April. I'll put the sign up link in the description, and we'll be looking at cancer mutations and actually touching on CRISPR-Cas9, which we touched on today. So we've got Matt Gahello, who'll be talking about his work in cancer mutations and how we can use CRISPR to, it's kind of similar to this, to work out exactly what we can knock out and change to hopefully treat cancer better. So if you missed any of this session, you'll get the recording link next week, along with those sort of further reading links. And yeah, thanks so much, Juliana, for joining us. And yeah, this is such an interesting talk. And thanks for everyone for joining us this evening.
Yeah, thank you for your questions. They're really interesting.